Great. So I guess we'll get started. So I'm Michael Passalacqua. I'm a postdoc at NYU, where I work with Joy Bergelson on uh, NLR genes, studying which plant immune hubs they interact with using single cell data, and a lot of co-expression. And today I'm going to be going through using ScanPy to do some data wrangling and uh, uh, kind of walk through what ScanPy is, how you can use it to interact with single cell data, some tricks that are helpful and things to watch out for when you're using ScanPy with single cell data. And hopefully by the end of this lecture, you'll be at least, familiar with ScanPy. You'll be familiar with what you can do with single cell data, and you'll avoid some common errors that people make when they're analyzing single cell data for the first time. So just to outline what we'll go through in the lecture before we move on to working with a Jupyter notebook, we're going to go through a little bit about RNA sequencing, a little bit about single cell data formatting and structure, so what the data will look like when you are working with it, and how you interact with that. We'll familiarize you guys with some common tools used for processing and exploring single cell data. And something I will really emphasize is the dimensionality and sparseness of single cell data and how that impacts the analysis and the techniques you use for working with it. So let's get started with a bit on RNA sequencing. So what is RNA sequencing? What is RNA? Why are we using this as our metric? So gene expression is a very important step on the path to establishing a cell type. mRNA is produced by transcribing DNA, and then it is typically spliced, exported from the nucleus, and then translated into protein. So proteins, which you think of things like enzymes or structural elements, define the function of what a cell does, and through that, its cell type. Cell types are kind of a tricky concept, but and ambiguously defined. But cells all share the same DNA, and despite that, there's a lot of different phenotypes of cells in the same organism with shared DNA, and they all work together to maintain homeostasis and to ensure the reproduction of that organism's DNA into its progeny. So we're really interested in cell types because they help to define what an organism is, what it does, and its phenotypes. So ideally, I think we'd like to be able to capture a protein and measure protein in every cell. That's really difficult. It's you can't really amplify protein like you can RNA. So RNA and DNA can both be amplified by being copied quite easily. In comparison, protein is much trickier. The molecules are also, there's 20 amino acids, but only 4 bases. So you also have a problem of right there. And because of that, we tend to use RNA because we can do much more high throughput experiments. We can capture a lot more samples. And even though it's not exactly what we want, mRNA does broadly correlate with protein levels, although there's certain cells and certain conditions where that doesn't happen. So what is RNA sequencing? So how do we count these RNA molecules that we're interested in? So RNA sequencing, our goal is to get the counts on the number of RNA molecules in a sample. In traditional bulk RNA sequencing, you take a bunch of cells. You might measure the number of counts of each of these different RNAs, and you'd map them back to the original genes they came from. And in a typical experiment, you might have some normal cells, and then you might have some mutated cells, and you might be comparing the expression of these different cells. Not only can you compare raw reads, but you can normalize these reads to the number of total reads, or you can compare reads relative to a certain gene. So you might see that, relative to the sodium channel gene, the DNA, there's very little potassium expression in this cell type. Whereas in these mutated cells, they have very high potassium expression relative to sodium. So there's a lot of things you can do comparing cell types based on their RNA sequencing results. We can actually take this further. We can then we can do single cell sequencing. So what we do is we have a bunch of cells in a buffer. They get mixed so that each cell is associated approximately one cell per bead. Once these fluids are combined, you then add a partitioning oil to separate out the droplets. So the oil surrounds the water that contains the cell and the bead. The cells then, this isn't perfect. So you'll have some droplets that have 0 cells. You'll have some cells, hopefully most of your droplets will have 1 cell. You'll have some other droplets that have 2 cells. So that's important to be aware of and know that that can impact the data you're seeing. Typically then, the cell will be lysed chemically, and then the RNA inside of it will be tagged by probes on top that are attached to the bead, and eventually then turned into a library and sequenced. So that lets us get a map of RNA data along with the cell that mRNA was from. So what do we do with this kind of data? And why do we do this? Because it seems like quite a bit of work versus just taking a bulk sample of cells. So bulk sequencing homogenizes the sample. It takes really the average of all the cells in it. So when you do this, you lose a lot of axes of information. You lose spatial information, so from the top of the sample or the bottom of the sample. You lose temporal information. So you don't know what age each individual cell is or how long the cell's been around. You lose cell-specific information. So if you have, maybe a trichome or a stomatocell, you're going to lose information about that versus its relationship to some cortex cells. And finally, you lose compositional information. So you don't know what percent of the sample is from one cell type and what percent is from a different cell type. And that kind of compositional information can be really important if you're looking at the level of protein or of a mRNA. You might imagine if you're interested in looking at whether a gene, is maybe provides pest resistance or something in 2 samples, one where it's higher or lower, you'd want to know if one sample it's higher because the cell type that makes that pest-resistant chemical, that pest-resistant gene is higher, or if it's because every cell is producing more of it. So that kind of compositional information can be really important. So droplet-based single-cell RNA-seq can retrieve all of these but spatial. So you do still lose spatial because you disassociate the sample into individual cells suspended in a buffer. There's spatial data. Spatial RNA sequencing is becoming more and more feasible, but it's still not quite all the way there in terms of data density. So we're going to be focusing today on single-cell RNA-seq. So what's the data you'll get from the single-cell experiment look like? So this is the AND data format. I kept this printed out next to my desk for like the first 2 years I was doing single-cell work because it's very useful to look over and refer to it. Something that's really important to mention is if you are using R and you're using Seurat, this data structure is the same, but it's transposed. So it's going to be reversed, which I'll explain what that is in a second. So in ScanPy, you have this .x matrix. So this is where your observations by variables are. So every square in this matrix is the level of a gene, which are variables, in a cell, which are observations. This could also be nuclei if you're doing single nuclei sequencing. So, and adjacent to this big matrix, you have 2 other matrices. So you have the observation matrix. So these are things about cells. So you might have the cell barcode, the cell's identity, what batch that cell came from, things like that. Above the matrix, you also have this other matrix, the variable data frame. So these are going to be your genes. So you're going to have, the gene names. You're going to have maybe how many cells that gene is expressed in. You might have you could have the average expression of that gene, things like that that are gene specific. There's also an added unstructured uh, dictionary that can just include other information about the um, single cell experiment, but most of your information is going to be in these three data frames. So if you're working in Seurat, this is going to be flipped. So your observations will be on top and your variables will be on the side of it. And this is kind of a very unfortunate difference between Seurat and ScanPy that it's good to be aware of. So what does the workflow for single cell data look like? So typically, what we're going to do is read in and clean the data set. This is important because it speeds up processing. You don't want to be handling a bunch of thousands of genes that are unexpressed. It also removes unused information that might clutter your analysis. These kind of very lowly expressed genes might have high dispersions, but very low expression. We're then going to QC check and visualize the data. This lets us check for major data handling errors. It can help us remove technical artifacts like batch effect, and it helps you get a deep appreciation for the structure of your data. So data can be, when you're doing data analysis in general, it's really important to just have a grasp of your data's what it looks like and generally. There's a pretty famous um, experiment, or I guess maybe worksheet that's done in a couple of universities where they uh, give a data set to two groups of students. The data set is shaped like a gorilla. If you plot it, just X and Y, it looks like a gorilla. If you just ask the students to like generate summary statistics like mean and whatever, mean and variance and standard deviation, most of the students do not discover the gorilla. But if you don't tell the students to do anything, you just ask them to explore the data set, almost all of them find the gorilla. So we would like to, for you to not miss gorillas in your data. That can be seen by just QC checking your data. And then finally, what we do is typically cluster and ID cell types. So what we're doing there is grouping grouping cells with other similar cells. This lets us reduce the data to human interpretable dimensionality. So it's very hard for people to envision more than 3D space. Typically, if you try and envision 9D space, you just think of 3D space and you say 9D in your head really strongly, and it doesn't really work quite well. Although if someone here can visualize 9D space, don't let me uh, be a downer. And it just also makes a lot of downstream analysis easy. A big reason why we do this with grouping similar cells is that single cell data is very sparse. So any one cell, we're not capturing all of the mRNA in that cell. We're only capturing a fraction. I think estimates for 10x based capturing estimate around 10 to 20% of the mRNAs get captured. Other methods like PIP-seq capture less, and then there's well-based sequencing that does capture more. But really, we do not get a perfect example of a perfect idea of what's in the cell. So it's really good to group these cells so that we can try and so we can reduce sparseness by combining similar cells that might be missing chunks of their expression profile. And we can also reduce, the other thing is that cells are not perfectly independent of each other. So these cells are all correlated. So the genes are correlated with each other because they're co-expressed into gene modules. So information from from two genes is not perfectly independent. So we can get mistaken statistical conclusions from over focusing on two genes that we think are independent, but really do have the same information contained in them, essentially. And the same is true for cells. Cells are, because they share cell types, are not independent of each other either. So trying to make conclusions on cell types when we don't have true independence can also lead us to make very vastly overstated statistical claims. A very common one is to compare two cell types using the number of cells as n, and that will give you a really inflated statistical estimate if you use some classical frequent statistics, and that can be lead you to just make false conclusions or make claims in papers that aren't true. So you really want to avoid that. So what are we trying to get? So this is typically what one of the first products of analyzing single cell data is. These are identified cell clusters on a UMAP. A UMAP is a dimensionality reduction technique. It's going to let you plot a very high dimensional data set, in this case, something with a PCA of maybe 50 dimensions in a 2D space. The reason I put this up is I want to really impress on everyone that you do not measure the UMAP. The UMAP is a very an early visualization tool. You should not make measurements in your UMAP. This is not a linear space, and any measurement between two locations on the space, that distance doesn't mean anything. It really is just to help you visualize clusters and to make sure that there's not any like tremendous batch effects in your data. Like if you tried to say, oh, the B cells are closer to the CD4 cells than the dendritic cells, that would be completely inaccurate. Do not measure in the UMAP space. You will get yourself in a lot of trouble. You will not make valid scientific conclusions. It's purely a visualization tool. That said, it's also not useless. There's some people who have very strong opinions about UMAPs, that they're useless or they're just reading entrails. I don't think that's true either. UMAPs are quite a useful tool, but they do have very, very strong limits, and it's very important to be aware of these limits and what you can and can't do with the UMAP. So please do not measure anything in UMAP space. But with that said, you can see how cell types that are similar cluster together and separately from other cell types. Now, I'd like to just share a couple of resources. I will put these in the chat as well for people to see. These are two really helpful resources for understanding high dimensional space and UMAPs. The first is lets you pick low dimensional data sets and then plot them with Disney, which is a dimensionality reduction technique that's pretty similar to UMAP. And it lets you see how the dimensionality reduction can really distort the space from something that's, completely understandable to something that's, looks like it might have structure but has no real structure. The second one is the Krishnaswamy's lab's workshop on using their dimensionality reduction technique, which is called, I think, Fate. And that dimensionality reduction technique is an alternative to UMAP and TISNE. They claim it keeps a bit more of the local structure of the data without having the problems of UMAPs where measurements can be completely distorted. I take that with a grain of salt, but I do think it's a really useful technique, and it's a very thorough workshop, so it's also quite useful, so I like to include it. Yeah. So with that kind of overall view of single cell data and what our goals are and some things to keep an eye on that you should be worried about, we're now going to go into a workbook. So I think it should have been in the chat for everyone. So if you click on that GitHub link, you'll be able to go to a Google Colab space. So I'll also be going through it and I'll be following through with it. So when you click on the link, I'll show you, let me share my screen so I can show you exactly what we're looking at. Let me just copy these links. Really quick into the chat. The first link, I'll also join it to forward on to everyone.
Just sent the first one.
Thank you so much. And now let me go ahead and share my screen.
And then now everybody should have the second one as well.
Great, thank you. Okay. So if everyone clicks on that first GitHub link, you should be at a page like this. What I'd like everyone to do is if you could click open in Colab, this will open a Jupyter notebook in a server run that Google hosts that lets you run some of the stuff without having to run it locally on your own machine. So I'll just give everyone a second to open that by clicking on that opening Colab link, and then we'll go through this Jupyter notebook to explain things. So I'll just give it one more sec and I'll check if there's any questions really quick. Okay. So what you just clicked on is a Google Colab link. What it did is it opened a Jupyter notebook through Google Colab, which is a free service from Google. A Jupyter Notebook is a coding tool that lets you run code in small blocks. This is really helpful, especially in data science, where you have to make a lot of small changes and you need to see results midway through. This is in contrast to something like a Python script, where you have to run the entire script all the way through, and it will then produce results at the end. So this is really helpful because you can make small changes to your code, see what the results are, continue on to a new code block once you're happy with one block. It has a lot of great visualization tools for displaying graphs and line in your code as well. That can make it a lot more understandable. Jupyter Notebooks also can be run not just on Google Colab, they can be run locally on your own machine. They can also be run on a remote server. So you can have, if you have a cluster, you can often run a Jupyter Notebook on that server and connect to it. A lot of IDEs will let you jump between headings and bookmarks in your Jupyter Notebook as well. So you can set it up quite nicely so you can have different sections with explanations. So Jupyter Notebooks are really useful tools. I use them to do almost all of my coding, especially for first passes before I convert things into full Python scripts I'm going to run on a cluster. But it's really important to keep in mind that there are a couple of drawbacks that can be quite major, especially in terms of reproducibility, which we're really focusing on for this workshop. So code blocks are executed in the order you run them. They're not deterministically run. So you can click run on a code block and it will run. You can run that code block multiple times and it will run again and again and again. So if you're not careful, they can also be run in an arbitrary order. So if you run a code block once, you can then jump to the end of the notebook, run a different a different code block, and then jump back to the start and run the first one. And those would just be executed in that order. So it's completely arbitrary based on where you click. So if you're not careful, you can end up making unintentional changes to your code without even realizing it by running something multiple times, running things out of order. This can lead to results where you don't know what's going on or you're not sure why something has happened. So it's really important that you always make sure you run your cells in the order in your notebook. It's good to frequently restart the notebook and run the code in order to make sure everything is running as it should in order, because otherwise you can have unreproducible results. And when you restart your notebook, it's important to know that the previous execution order is lost. So you won't know what order you did something in the past. So it's really important to run the cells in the order there in your notebook so that your code works as you expect it to do. So before we get started in the notebook, I'm just going to show you guys a couple of things you can do with Markdown, which can help you make the notebook more readable. So if you double click on any of these blocks, you'll be able to see the original Markdown code on the left and the output on the right. So you'll see I double clicked on this to open this up, and you'll see here's what I typed, and here's how it's rendered. You'll see that we have different heading sizes available. These can often be jumped to in the notebook. I think Google Colab has, headings, a table of contents on the left that you can see over here. And those are pulled from your Markdown headings. So these are created by just putting a number, a pound sign at the start of the line, so you'll see we have Markdown heading 1, creates a large one. If we put multiple pound signs, it still makes a heading, but it becomes smaller. So we have 2 and then 3 here. There's a lot of things you can do with the text as well. So you can bold text by putting double asterisks around it, as you can see right here, where I put two asterisks around bolded text. You can italicize text with a single asterisk. So you can see here, italicizes a single asterisk around it. And you can do basically almost any text or table formatting. You can do strike throughs, you can do a lot of useful stuff for just making the notebook more readable. We have a question in Q&A. I think that will be shared later, the video links for this. So yeah, so that's how you can create Markdown code. You'll also see you can put lines into the code into Markdown. So I put a line here. So 3 dashes under right here becomes this line when it's rendered. So that can also be quite useful to break up code and make it easier to read. So below that Markdown block, we've actually created a code block. So you'll see that in this development environment, when we can either click add a code cell or add a text cell. So Markdown cells are text. Code cells are Python code here. In this case, I will point out that Jupyter Notebooks can also be used to run R code, and so it's just whichever you're more comfortable with. So they're quite useful for both. So in Python, we're commenting off these lines with a number sign. So that lets us just make comments that won't run as code. And below, what we're doing is we're using this magic exclamation mark at the start of the line. That tells the Jupyter Notebook that it's going to run these as shell commands. They're not going to be run in Python. So we're actually running these as shell commands. So what we're doing is we're installing ScanPy. We're going to install an algorithm for ScanPy that lets it cluster things. We're also going to make a data directory. Then we're going to fetch your data, and then we're going to make a writing directory. So if you just hit run over here on the left, that will run the cell, or you can hit Shift Enter, and that will run it. So this is not authored by Google. This is authored by a random GitHub, which is our random GitHub, and it will. But so if you just click Run Anyway, it'll run it. So here we're going to install ScanPy. So this is going to install quite a bunch of things. So you'll see these are all the packages and dependencies ScanPy uses to run. So if you don't mind, it'd be helpful if everyone was also running this alongside so that you guys can play with the data. See, we also downloaded this data set that we're going to use. This is the same data set that was generated from Andrew yesterday. So now we're going to go to Python code. So we're going to start off just by importing all the packages we're going to be using today. So that should just run. Okay, so now we're going to do a couple of things that are really important for reproducibility. So first, we're going to set up our random seeds. So a lot of processes in single cell data initialize randomly. So to ensure that we can reproduce these results, we're going to set the random state, and we're going to use that in our code. Once you have your results and you're done with your code, it's often good to try using different random states to make sure that your results are robust to different random states. It's quite rare to find results where they're sensitive to the initial random state because a lot of these processes we start where we initialize randomly the goal is that they're trying to find is like a stable state. So they tend to find that state no matter where you start from, but it is possible. So it's important to make sure that your results are robust to those changes. So here we're going to set our random seed. That's just any number you can pick as your random seed. And we'll just set that for both NumPy and our random package, and we'll set it later as well for ScanPy. So here, we're also going to set some thresholds up front that we're going to be using. We define them early so that we tell someone who's reading our notebook that we have assumptions that are not buried deep in our code because notebooks can get quite long or quite dense, so it's good to have them up front. It also lets you change them easily without having to search the whole notebook. So if you really want to change them easily, even easier, what you could do is you can make a new text block. You could put a heading here, and you could put uh, threshold definitions. That would show up. And then when we look at our outline, we can now pop over to click it to pop to threshold definitions, which is quite useful. So here we define some thresholds. We're defining how many genes we want in every cell for us to use that cell in our single cell data. We're asking how many cells a gene has to be expressed in for us to be interested in it. The max number of different genes per cell. Here we're using the max percent of mitochondrial genes. We're also defining some things for highly variable gene selection. So these thresholds, and then some clustering parameters. So we're going to run these cells. We're also going to set some settings for ScanPy. So here we're going to tell ScanPy that we want it to be chatty and inform us of what's going on. So this is a verbosity setting. We're also going to set some figure parameters. So we're going to ask for a white background and a figure size of 5 inches by 5 inches. This helps a lot when we generate PNGs to ensure that they're on a white background, not transparent. And then we're going to just read in our data. So here we're reading in that data set we downloaded earlier. And we're these drop columns just remove some empty columns that are in the data set. So if you've worked with genetic data a lot before, you can know that some gene symbols can overlap, especially if they're short 3-letter gene symbols. So we would like to make sure that that does not impact our analysis. So we're going to ask ScanPy to make these gene names unique. So we just use this .var make unique names. So if there's any duplicate variable names, we'll add a number to it to separate them out because that can otherwise impact your data. And we're going to explore the structure of our data a bit. So if we just run just a data, like our data structure and data, we can see a little bit about it. So we'll see that's an object. We have observations and variables. So if you recall from earlier, those two side columns you mentioned. And we also can see the size of our main object, which is our results of single cell data expression experiment. And we'll see that we have 2,269 OBS. So OBS are our cells. So we have 2,269 cells. We have 3,000 or 30,000, 31,000 genes approximately. We can also see that the OBS data frame only has one column, sample, and the variables. So the cell data frame only has sample, and the variable data frame only has gene symbols. We have a question in the Q&A. So you don't need to set a working directory. So this will, if you just run the code up here, it basically will set the working directory for you when we, and all of the code is set for it to write to the correct place. So we can now view our AND object. So if we we're asking for the code here to do is we're asking to print the type of the AND object. So just to see what it is, we're going to display it just to view it, and then we're going to also print out the unique parts of the column. So what do we see here? So we asked for the type. So this is a pandas data frame. So if you've worked with Python before, you're probably familiar with this data structure. A data frame is basically a large table that has a lot of useful functions built into it. So we can see that our observations part of our object is a data frame. So you can manipulate just like you would a data frame. Anything you do with the data you would normally do with the data frame, you could do to the observations part of the AND data completely normally. We can then look at it. We can see we have our index here, which is our, you see these list of letters. These are our cell barcodes. And we have also some information about where this came from. And you'll see that we have our sample, which sample it came from. So you can see some of these came from, these came from different samples. If we then zoom in on these samples with unique to ask what are the different things here in the sample, column, we'll see that we have uh, four different sample types. We have two control reps and we have two experimental reps. We can do the same with our variables. So if you remember, so observations, if you remember, are cells. That's why they have cell barcodes. Let's look at our variables. You'll see that the variables are genes. So here we have both our gene IDs and our gene symbols. So we have our full or adopted gene names and then also their shorter names that are a bit more readable for certain things. And then finally, let's get to the meat and potatoes of this object, which is the big .x in the middle of it. So this is the data matrix of our indata object. So this is going to contain our, so this is our results. So we'll see it's a sparse matrix. So unlike the other two, which are data frames, this is a sparse matrix, which can be, might not be a object you're familiar with. And you'll also see that's quite large. So we have, 70, so we have the shape of it right here. So, like I mentioned earlier, single cell experiments are quite sparse. Because a single cell experiment has about 30,000 variables for each cell barcode, the matrix is quite huge, becomes huge as you add cells to it because every single cell you add adds 30,000 possible variables. However, most cells do not have reads for most genes. So our matrix is quite sparse. Because of this, we can just skip storing zeros. So instead, we can just assume a zero is everywhere. And for every variable, everywhere there's not a zero, we can just say where there is something that's not a zero. So this sparse matrix is a lot more computationally efficient, and it can manage a lot of this behind the scenes for us. We do need to keep this in mind for some things that require dense matrices, certain ways we write code or certain loops. For a lot of NumPy operations, this can just get handled in the background, but certain things you'll do, especially with pandas, might not like using this dense matrix. So let's just look for some information about this experiment and look at how sparse this matrix is. So in the past, I've used an older data set to do this, and it was really quite sparse. With better chemistry for single cell experiments, these experiments are a lot less sparse nowadays than they used to be, but we'll still take a look at how at this matrix. So here, what we're going to do is we're going to make get a dense version of this matrix. So we're going to fill in all the zeros, and then we're going to view it. If we view this matrix, so what you'll see if you run this code is a lot of zeros. There's definitely some values in here, ones and fours. You can see that the overwhelming majority are zeros. We can then view the dimensions of the matrix really quickly. So we'll see, as we expect, it's 269, which is the number of cells we have, and 31,000, which is the number of variables we have. So that's all as we expect. So that's just a good sanity check. And now what we're going to run is this code block right here. And what this code block will do is going to calculate the number of values in the matrix. So you just get the length and width of the matrix. You multiply that to get the total number of values. We then check which positions are non-zero. So we ask for where are there non-zeros. We sum those to get the total number of non-zero positions. And then we just divide this to get the percent of our matrix that is. And what we see is that actually we only have a value at 11% of our position. So 90%, 98%, 99% of, excuse me, 89% of our matrix is completely empty. So yeah, so it's really quite sparse. So that's, something that influences these dimensionality reduction techniques, these and these clustering techniques that we're going to do. Okay, so that's some good information about our matrix. So let's now look at our actual data. So what we're going to do is plot highest expressed genes. This is a built-in ScanPy function. It's going to let us visualize, it's going to show us what genes have the most reads. This is really good, useful for sanity checking. So let's just take a look at what we see when we run this. So you'll see is that we have this list of the top 20 genes that are expressed in our cells, and you'll see that one of them is actually quite a bit higher. So we're going to take a second to explore our database our data set because we do want to have a good understanding of what's going on in it. So we're going to go ahead and look up this gene AT1G0759. We'll do it together. So we have this gene right here. We're going to look it up. So there's a lot of useful resources for looking up Arabidopsis genes. So we're going to look at SUBA, which gives us its localization. And what we're going to see is that this gene is really heavily expressed in the mitochondria. You'll see that its subcellular localization is all in the mitochondria, basically. So SUBA is a really useful tool for seeing where these genes are expressed, where genes are expressed. And if you're familiar with single cell data at all, you might know something about what a highly expressed mitochondrial gene might indicate. We'll take a minute or two if anyone could take a second and pick a gene from a different gene from the data set. If you would just paste into Google and take a look at maybe SUBA, or you could look at TER or uh, University of Toronto site. They're all really useful um, resources for understanding the gene and what it might be. UniProt's also quite helpful. But yeah. So while everyone does that, I'll also just mention a bit about the structure of this graph. So if you look at this graph, you should think about what each dot on this graph is. So, this is our distribution of cells. So we have the line here is the median. These dots are outliers. Every individual dot is a cell, and it's the percent of counts that are present here. So for like this cell, it has 4% of its total counts are from this gene here. So you have quite a few cells with very high expression of this mitochondrial gene. And we'll get into what that means in a little bit later. So, like I mentioned earlier, it's really important to have a broad understanding of your data set. So this lets us see if anything went dramatically wrong. So if you saw all of your most expressed genes as stress genes, you might be concerned about that you mishandled these cells prior to sequencing. There's lists of protoplasting-induced genes. So if those are really high, you'd also probably be a bit worried. And mitochondrial genes, you'd also be worried which we'll get into in a little bit. So in a normal workflow, if we had, you might also do background detection here. So when you're doing single cell sequencing, RNA has a tendency to leak out of the cell's nuclei and into the ambient fluid. That ambient RNA can then get mixed with every cell's profile. This can be really problematic for marker detection, especially where if you detect a gene broadly, you might rule it out as a marker, but it can be caused by one cell type just leaking that RNA into the background. For this kind of, so if you're going to do this kind of detection, which is typically run from a separate program, I'd recommend something like, from the terminal, I'd recommend something like CellBinder or SoupX. CellBinder is a lot more computationally intensive. It has a little bit better results typically, but it can take quite a bit longer to run. I think we have a quick question in the Q&A. The number after AT. So the number after AT is the chromosome number for Arabidopsis, which I think someone was also typing out an answer, which is good for it to be in the chat permanently. There's also another question that's if you're running comparable pipelines in Seurat and Scampi, how reproducible are the resulting embeddings and cluster assignments across frameworks? That's a really great question. There's actually a couple of manuscripts on this exact question. Typically, the cluster assignments are quite reproducible. The exact embeddings are in the PCA space tend to be pretty reproducible. There's a couple of issues with how Scampi and Seurat handle log transforming data that I think have been corrected now. But there are, if you search this, search it. There are papers on this topic, which in the past they were less reproducible than they are now, which is good. If any of the other panelists know the paper I'm referring to, it'd be great if you could reply to it to that question with the link. So now, let's say we've done our background detection as well. We're going to keep going, and we're going to remove some cells and genes that we're not interested in. For genes, we're only interested in genes that are expressed in at least a few cells, because a gene in a single cell doesn't really tell us anything. We're also interested in cells with enough reads that we can cluster them. And something else that we might want to do is remove genes with a high mitochondrial signal, like for example, from that first gene, as this can indicate that the cells are dying or are low quality. So when cells enter apoptosis, there's a big breakdown in the mitochondrial in the mitochondria that helps trigger apoptosis, and that a lot of reads from mitochondria can indicate that. So here we're just going to filter our cells. So here we're just going to filter out ones that have cells with less than that threshold we set earlier, and cells with the less filter genes that are less than the threshold we set earlier. So let's run that. ScanPy is going to tell us how many things we filtered out. So we only filtered out 181 cells, but we have filtered out over 10,000 genes. So you can really think about how much this is going to cut down on the size of our data set by cutting out 10,000 genes from 30,000 to around 20,000. So now let's explore this clean data set. So let's look at our observations. And what you can see is that we have now only 2,088 rows. I'm just going to make a quick change to this, where we can also look at our general things. We have 2,088 rows. We have only 20,000 columns now of genes. So we've really reduced our data set quite a bit to make it less computationally intensive to run. What you also might notice is that previously we only had sample and gene symbols listed for our object. We now also have number of genes and number of cells. So now for every cell, we have a column that says how many genes are expressed. And for every gene, we have a description of how many cells that gene is expressed in. What we're going to do now is annotate that gene that we identified earlier as a mitochondrial gene, so that we can remove cells that have very high expression of it. So here we're just asking, we're just setting a column MT, and then if we view that change, you can see is now we have, in addition to that number of cells I mentioned earlier, we now have this mitochondria column for every gene. For that one gene, so this column is for every gene. For the one gene what we think that we identified as mitochondrial, that's going to have a true value for it. Now we're going to do some QC metrics just to view some things. So we can calculate a bunch of QC metrics, and we can also feed it specific lists of genes that we'd like to have information on. So here we're asking for mitochondrial. And we'll see that this also changes our database quite a bit. So now when we look at the observations, instead of just sample and number of genes, we've added number of genes by counts, the total counts, the total counts of mitochondrial reads, and the percent of counts that are mitochondrial. So we've really added quite a bit of data with just this command. So now we're going to, just to explain what all these are, why the number of genes and the number of genes by counts is different. So this is different because we've now filtered some genes out, and this is only going to show ones that we haven't filtered. Which is a very common question people have. So let's just visualize this. So we're going to plot some violin plots of all three of these. So we're using the data built-in ScanPy's built-in plotting functions. So every dot here is going to be a cell, and the violin plot is our overall distribution. So what you'll see is that we have genes by counts, mitochondrial percent on the right, genes by count on the left, and total counts in the middle. So what do we want to do with this information? So this can help you do a lot of filtering. So what we would like to do is filter genes with too many genes by counts. So that's too many different genes expressed. We have a suspicion that these are that these are actually droplets that got more than one cell, or some other technical artifact. We can also remove cells that have too much mitochondrial DNA based on this percent counts of mitochondrial over here, as we think these cells are dying and are low quality. As an alternative to thresholding-based approaches, you can also use for doublet detection. So for the number of genes by count, you can also use doublet detectors like doublet finder or doublet detection. I would recommend typically nowadays using a doublet detector. Uh, when single-cell RNA was newer, people typically just set thresholds, but now I'd recommend doing a doublet detector. So let's do this filtering. So here we're just going to slice our matrix. You'll notice that we're just using normal matrix, normal array slicing in Python to cut this and add object down. So we're removing cells with more than 2,500 different genes in the cell, just as largely just to reduce the size of our data set to make it a little bit easier to plot. But also, we're going to say that those are we think those might be doublets. We're also going to remove cells with more than 5% mitochondrial reads because this actually should say 1% mitochondrial reads because we think that those are low quality. Those 5%. So now that we've finished filtering, we're going to save our object. So you might say, well, we're not done with this notebook. Why are we saving an object here? It's really important when you're using Jupyter notebooks to save intermediate objects in case you have an issue with your pipeline that you need to fix later, or you have analysis without running the entire thing. Changes to packages can also happen that break code, and you don't really want to spend days getting it to work again when you're in review and trying to get something turned around quickly. So saving this can really help reproducibility a lot. It can help you run code faster. And if you made an error with your running the order of your script, this can also help you catch that kind of a thing as well. So this is a really important reproducibility to have routine checkpoints as you go through your code. So you're going to write our filtered database down. I'm not converting strings to categories here, but depending on your data frame, that can save a lot of space. So just so you know that this is an option. So now we're just going to normalize the data in logarithm and log transform it. So first we normalize it. So we're scaling the count in each cell to sum to 10,000 so that we can make comparisons between cells easier. We're then going to log transform the data. So we add 1 to the data and then log transform it. This helps us deal with zeros because there's no log 0. And now what we're going to do is we're going to start working identifying cell clusters. So this is often kind of the part of the single cell sequencing pipeline people are most interested in because it's quite important. And identifying those clusters lets you make comparisons and analysis between them. So to make clusters, the first thing we need to do is identify highly variable cells. If a gene is highly variable genes, if a gene is expressed at the same level in every cell, obviously it's not going to help us distinguish two cells. So instead, we need to find the genes that are the most different between our cells. So we're going to run this. It's going to use the thresholds we set earlier at the start of the data frame to identify our highly variable genes based on their expression and their dispersion. So this is going to add information to our .var because this is information about genes. So it goes in the .var, our .variables. So let's take a look at that, and you can see that we now have quite a bit more information. So we have mean counts, we have total counts, we also have a flag if it's highly variable, we have the means of that gene's expression, we have the dispersion for that gene, the normalized dispersion for that gene. So you have quite a lot of additional information here that's going to be used to identify variable genes. Okay, but what does this look like? So let's just plot our variable genes. So ScanPy has conveniently a built-in function for visualizing these highly variable genes that we've identified. So here we're going to take a look at our dispersion So this is our normalized dispersion and our expression of these genes. What you're going to see is that in black are highly variable genes. Um, you don't want your all of your genes to be marked as highly variable genes. You really do want to identify a limited number of highly variable genes. Um, Typically around like 5,000 or less is about right for highly variable genes, but can depend quite a bit on your data set. So that's something that you can play around with and see how it affects your results when you're doing analysis. So now we've found our highly variable genes. Let's reduce the dimensionality of our data uh, uh, with PCA. So I'm going to explain PCA kind of briefly, but we'll skim over nearest neighbors because it's pretty intuitive, is you're finding things that are nearby in the high dimensional space. So what is PCA? So PCA is basically finding the axes of variation that make them that are the largest, and setting these to and rotating your data to match these axes. You can imagine if you had a data set where things varied along x and y equally, x and y both don't provide any information separate from each other, because if you know x, you know y. So if you could then rotate that whole data frame so that that whole data set so that one of its axes was the combined x and y, and that would give you an axis of variation. PCA is finding those, doing that repeatedly to find the top axes of variation. So the first PCA is the combined factor that explains the most about the variation in the data set. The second PCA explains the second most, so on and so forth, until for however many PCAs you calculate. The most number of PCAs you can calculate is the number of uh, uh, either observations or variables you have. You can only calculate as many PCs as basically as you have either genes or cells, whichever is lower. But typically we're going to calculate a lot less than that. So PCA also uses a random state. So just to point that out, we're using that here. So we're going to do our PCA calculation. You can uncomment this if you're interested, just to show what PCAs contribute to variance. You can uncomment if you're curious and like to see that. It won't affect the code. So now we're going to calculate our nearest neighbors for our 40 principal components that we calculated from PCA. We're going to find the nearest neighbors for every cell. We're going to figure out which cells are near which other cells in the PCA space. We're then going to reduce that with with a dimensionality reduction to a UMAP. So this is going to be a 40-dimensional space. So just to explain where we've gone, we went from a 20,000 dimension space of 20,000 genes. We've reduced it to a 40-dimension space with PCA. And when we do a UMAP, we're going to reduce that down to a 2-dimensional space to view it. So we're going to plot our UMAP. So here you'll notice we also have set a random state to ensure reproducibility. We're going to cluster that. So we're going to use a Lagan algorithm to cluster this. It's going to run quite quickly because we don't have very much to cluster. So it's going to cluster them, and it's going to use the distance in the multidimensional space, so in the PCA space, not in the 2D UMAP space. The clustering does not occur in the UMAP space. As I mentioned before, UMAP space is not really reliable. UMAP is a projection of a high dimensional object to 2D. You can think of it like a shadow of a data set. So it's going to have distortions just like a shadow would. You can see in our observations, we've now added the cluster, which is under Leiden. So these are the cluster assignments for everything. And we're going to visualize our clustering. So here's our UMAP. So it's our 2D space. You can see that we have a couple of clusters identified. The reason I picked this specific clustering is because it does highlight how the clustering happens in a multidimensional space. So you have these brown cells here and here assigned to the same cluster, even though they're separate in the UMAP. That's because UMAP is not a true rendition of the high dimensional space. So these are closer, even though that they're closer to each other than they are to these green cells, even though in the UMAP they look closer to the green cells. So that's just a great example of why you can't measure distances in UMAPs. Okay, so we're going to take a couple of minutes to just play with the clustering settings. You can change, you could try changing resolution. You can try changing the number of neighbors or the number of PCA one at a time and see how it changes the data. You can combine them. You can also try changing the highly variable gene selection cutoffs and any of those changes, and you'll see that it'll change the UMAP quite a bit. Yeah. So I guess we'll probably let people do that on their own time since it's a bit awkward to sit here for 10 minutes while people play around. But just to give an example, I'll change it really quickly just to show how UMAP can change. So we're going to change only the random state. So we're going to change it temporarily to a different random state just to see how the UMAP changes. And you'll see that we have a different UMAP. So the random state does change, but what we'd expect is to see that most of these cells are still assigned to clusters with the same cells, even though the exact projection of the UMAP has changed. And you'll see that in this projection, that the brown cells actually did end up together. So that can, that's just a really great example of how changes in the UMAP space can change and not really reflect reality in any way. And you'll see that when we put the variable, restore our original random state, we go back to the same random state we had previously. So that's just to show that it is reproducible as long as you set the random state. Okay, so now again, we're going to save this. So we just did something that's kind of random state dependent. It's good to save here just to get an idea of, just to make sure that we can go restore our progress from this point in case something goes wrong later. Okay, so now we're at what everyone's really interested in, which is identifying clusters. So here we're going to use 3 markers for root cortex cells. So what we're going to do is we're going to plot the UMAP, and we're going to add, on the UMAP, we're going to tell it to plot 4 things. We're going to plot the original clusters, Leiden, by telling it the Leiden column. And we're going to give it 3 gene names, these are cortex cell markers. And we're going to ask it to plot the colors as the intensity of expression of these 3 markers. Let's go ahead and plot this. Cool. So what do we see? So on the left, we have the UMAP from above. So you'll see it's the same UMAP with our Leiden clusters or our cluster assignments. Then we have 3 marker genes. So you'll see that for each marker gene, so these are root cortex markers, we have expression, which is yellow is higher expression than blue or than dark blue, which is 0. So the more yellow or green it is, the higher expression is. You'll see for all 3 of these markers, they're quite highly expressed in this red cluster here, cluster 3. But this is a great example because you do see leaky expression in other cell types, even though these are cortex markers. Excuse me. You do see expression in other cell types. So it's important to know that cell types, so expression markers like this are often not perfect. So even though they're very highly expressed in one cell type, they might be expressed at a lower level in a different cell type. And you'll see that different markers have different amounts of leakiness. So like this one is pretty tight to the red cluster, whereas this one you have quite a bit of expression down here in these blue and green clusters. You'll also notice is that even within the red cluster, there's different expression of these markers. So like this one is maybe more expressed on the outside of it, whereas this one is expressed more maybe in these in the middle part of the red cluster. So clusters, might have heterogeneity in them. Clusters are not perfect. And we set the resolution. So we told it how many clusters to make, quite low. So, that can all those kind of thresholds you set do change things. So if you like, I have a block here for you to put genes that you're interested in on your own. You can take genes from above, where we looked at the most expressed genes, or you can pick genes that you know and like, and you can plot a couple of genes on your own just to see what it would look like. So as long as you just put the gene name where these numbers are, it'll plot that. So here I just want to show you guys some metadata. So in addition to the OBS column, there's also an OBS M column, which is OBS metadata. And you'll see that that's where some of this data was saved. So here we have our PCA vectors and our UMAP vectors were embedded here. So if you need to plot something on your own with the UMAP vectors, you can do that with this. Great. So now we've identified one cluster. So we're going to label that. So we're going to set our dictionary just with this cluster relabeling. We're going to in the ligand column, we're going to replace the 3 with cortex, because based on this expression pattern, we think the red love thing here is cortex. We'll run that. And this is just a warning that, there's been a pandas update, so we need to change how we're going to write this kind of code in the future. So let's plot this out. So here, this is the same UMAP function, plot UMAP, we used earlier, but we used a really, I find, useful plotting variable where we tell the legend location to be on the data. So let's just put our labels directly on the data. And what you'll see, sorry, it's a little small, is that now cortex has been labeled directly on the data, and the other ones still have their original labels. So now we can see we have this cortex. So if you were working with this data, you'd probably use more genes than 3 to identify their cell types, but you would do that repeatedly for every cell type until you had identified all of them. Nowadays, there's transfer learning approaches to use existing labeled data sets to label new data sets. I think in plants, they don't work nearly as well as they do in mammals, because plant cells are not as terminally differentiated as mammals. But so that can be a challenge. But it is an option to consider if you don't have a good idea of what these cell types are. Okay, so we have clusters. Now this is typically when you do things like you could find marker genes, you could ID cell types, you could do differential gene expression between clusters, and do all of that kind of fun stuff. So this is kind of like, one of the main products you're trying to generate when you're doing single cell. And now that we've gotten here, there's one more quick thing we have to put Q&A. Let's see. Yeah, of course. So one thing that I'd like to go through is pseudo bulking data. So I think a very, very common issue, I mentioned it earlier, is that people will make mistakes in single cell, is that they will treat every cell individually. So pseudo bulking can help us handle the sparsity of single cell data while also being more intuitive to handle statistically. So if you want to compare 2 cell types, you might think of each cell as a sample and do your statistics that way, but that would give you dramatically more confidence in your estimate than is real. Because cells in a cluster are not independent. They're put together because we think they are similar based on their expression. So treating them as independent is not really statistically accurate. So by pseudobulking, we can instead reduce sparsity and by combining cells, and we can also reduce the cells to true independent samples based on batch. So we're going to create some fake batches. We do actually have real batches, but the names are quite long and it makes the code a little harder to read. So we're going to assign fake batches to everything. So here we're assigning random batches. You'll see low 1, high 4. So we're actually going to make only 3 batches, but it's going to look like 4 batches because of Python. And what you'll see is that we've now added a batch column right here on the right. So that column is now our fake batches. So we have 3, 2, and 1. So every cell is been assigned a batch. So we don't have 4, even though it says high 4, is because Python is inclusive on the low end, but not on the high end. So it's basically saying under 4. So we're going to plot these. So this is also something, a really important thing to notice when you're plotting in ScanPy. So here we have the batches are a number, right? So they're the batch is an integer. So ScanPy thinks this is a value about the cells. So when ScanPy plots this, it plots the higher values on top. So what you see is that it looks like we have way more cells in batch 3 than we have in batch 2 or batch 1 because it's been plotted as an integer. So this is called overplotting. So this makes it look like we're dominated by batch 3, even though that's not true because we know we just randomly assigned everything, so they should all be even. This can really trap a lot of people. It can lead to people making figures where they assume one cell type or one gene or something is expressed much higher than it is. When we're going to change this batch type to string, and we're going to plot this as, again, with this as a string. And what you'll see is that our overplotting is resolved. So now 3, which is green, looks just as common as 2 and 1. So this kind of overplotting is really, really important to be aware of. Uh, uh, and you should really be careful because that can lead you to make completely wrong conclusions about expression or about about batch variables or about something being more enriched somewhere that's not true. So this is really, really important to know that overplotting happens in ScanPy. This is actually not a problem as much in Seurat, I think, based on some of my co-panelists who use Seurat more than me have said, where Seurat automatically prevents overplotting, but ScanPy does not. So it's good to know. Okay, so now we're going to loop through the batches and build a data frame. This code is going to assume all clusters contain all batches. This is might not be true in all data, where some batches might have not be missing a rare cell type, but just want to make that clear. Um, uh, so so here we're just going to get our cluster names. We're going to get our batch IDs. We're going to generate a list of all combinations of batch and cluster. We're going to just look at our combinations. So we have, these are our clusters on the left. You can see some of our cortex, and then on the right are our batches. We're going to make a data frame that we're going to attach things on. So these are our genes. We're going to loop through every batch, and we're going to pseudobulk it. So basically for every batch and every cluster, we're going to average the expression of all genes in that cluster, and then we're going to add it to our data frame. We're then going to write this out. We're going to write our final object out. This is our final PBMC object, or our final single cell object, just to make sure that we have it, because we're at the end of it. And then we'll view our pseudobulk data frame. And you'll see that for cluster number 4, batch number 3, we have our averaged expression. We have our average expression. And we have this for every cluster number and every batch. So we have 21 combinations. So we have 21 pseudobulk. So if I was going to do differential expression, I would then make comparisons between clusters with the batches as independent reps. So I'd say, for cluster 4, we have 3 reps. For the cortex, we have 3 reps. And I can do comparisons between those. You'll also note that even with pseudobulking, we still have differences between batches, even though this is completely random. That gives you an idea of just variation there is between individual single cells, which are very noisy and can be quite messy. Yeah, so I think with that, we're done with this notebook, and I think we'll probably go to some Q&A with everyone. We have a couple of Q&A. So there's a question, what's the end result we expect from single cell, UMAP, differentially expressed genes cell-wise? So, I mean, it depends on what your goal is. For often, it's differentially expressed genes between clusters. That's a common one. The UMAP on its own isn't really useful for much other than as a check. So really, often the end result is either marker genes or it's maybe differentially expressed genes, but it just depends on why you're doing the experiment. And it is marker genes for cell annotation. So there are automated cell annotation methods in plants. They don't work as well. They're often called transfer learning approaches. But there are automated methods that do work. It's just you have to be careful to make sure they're good. I think Ben also wants to chime in.
on this topic?
Oh, sorry. It just said on the Q&A, it said Ben Cole wants
Oh no,
to.
I just saw that you were answering it live.
Okay, sorry. Um, yeah, so I think for that it does. I mean, yeah, the goal depends on what you're trying to do, but yeah, there are transfer learning approaches. I would be just very careful with them in plants. Plants are less terminally differentiated than mammalian cells, where these are often developed. They're often, in fact, developed in the immune system, where you have very terminally differentiated cells. And because of that, they have very, very reproducible expression patterns that are less common in plants. This extends to UMAPs, where plant data tends to look more smeared than or more less well clustered than mammalian data because plants are not terminally differentiated. If you take plant cells, you can regenerate them into a whole plant. You can't do that with human skin cells yet, but that terminal differentiation does affect things in plants. It makes it quite a bit more difficult to work with plant data than regular data.
Yeah, I should have also mentioned yesterday, which I think I failed to do, that in the Google Drive I shared, we do have a spreadsheet of marker genes based on the Arabidopsis root atlas that Rachel Shahan produced when she was at Phil Benfey's lab. So that those can be used to kind of help explore the data that we provided. I think, Sharon, you probably remember what, I think there were only 4 cell types that we included in this. So don't expect this to be a very representative data set.
Yeah, it's a very slimmed down data set. The question is if we have any tutorials and for learning about Seurat and R. Seurat has a pretty good intro tutorial on its documentation page. I don't know about any more detailed ones. I don't know if I use mostly ScanPy, but maybe one of the other panelists might have some suggestions for Seurat tutorials specifically. But I'm not super familiar with any of them beyond just the basic Seurat tutorial.
Yeah, I think the vignettes there are a good place to go.
Yeah, I just put a link to the vignette page for Seurat in the Q&A.
Okay, one more question. Mutant changes cell identity happens to the same cell type across genotypes. That's a very great question. Uh, so when you have if a mutant is changing cell identity, typically that would be a separate batch. So that would not be suitable with the same cell type within uh, across genotypes. You'd pseudobulk only within batches, and that would separate out your mutant or your experimental condition from your control conditions. If you have different genotypes, like let's say you have uh, as long as you're aligning to the same reference genome, it shouldn't be an issue. If you're aligning to a different reference genome, then you're getting into like cross species that can get really tricky. And there's a couple of approaches you can take. I've published some approaches on using co-expression for orthology, for aligning orthology across species, but you might just use orthogroups instead. What are the gaps you see in Arabidopsis or rice single cell, or any specific rice single cell atlases like Arabidopsis? I think there are some rice single cell root atlases now. I think there's one of the quite early single cell papers was actually, I think, root for Indica and Japonica, if I remember right. So there are that. Gaps, I think the main gaps in literature right now are in conditions specific expression for single cell. We have quite a few atlases, but we don't have a lot of condition specific. All that is being rapidly addressed. I think actually a big gap is using single cell in a targeted way instead of just atlases, which is also starting to be addressed. Those are great questions. How can I import a Seurat RDS file to analyze with ScanPy? Great question. This is something I do a lot because I work with a lot of have a lot of collaborations, and some people prefer working in R, some people prefer working in ScanPy. You can export a Seurat file to a .loom or to a .hdf5. There's using Seurat disk or Seurat. I forgot what the other one is, but I think if you search Seurat disk, I think it should come up. And once you've exported them to a Loom or HDF5 format, you can then read that into ScanPy quite easily. So that's how you import a Seurat RDS file. So you'd save it as a Loom or HDF5 file format. Next question is cell marker data consistent across different analysis pipelines or tools? Great question. A lot of people would say cell markers are completely independent and should be fine. I think you can have impact, especially for nuclei data, where you can have certain cell markers that don't work as well in cells as they work in nuclei, or work better in nuclei than in cells. Other than that, I think marker data tends to be pretty good across different analysis pipelines. It's just when you have different modalities where marker data can really have issues, I would say. Okay. How can we connect with you guys? You can email me. My email's michael.p@nyu.edu, which I think is on the PCA like flyer or something. Or you can I don't know, add me on Twitter or something.
Hey, Michael, can you talk more about what you can do with the Jupyter Notebook format? Like, can you like in terms of generating plots that you can publish, or publishing like an entire output from the notebook, reproducing things across different platforms, different like outside of Colab, that kind of thing.
Yeah, so notebooks are their own files. So the notebook contains both your output. So if I saved this notebook we're looking at right now, it would contain all these plots we've generated as image files. It would also contain as images. It would also have all our outputs like this in the notebook as well. So that lets you, if you're sending someone a workflow, you can send them the whole notebook, which is really quite useful. You can also, in addition to just displaying outputs like this, you can do things like plot.savefig just to, if you have um, um, to be able to save these outputs to specify to image files so that you have those separately that you can include in the publication. This is really useful for when you're generating image, especially for publication image generation, because you often have to iterate, a dozen times to really perfectly lay out the entire image, the entire plot. So you can do that and then save it with matplotlib, and the same in R with ggplot. You can also do the same kind of iterative generation of an image. Was there anything else, Ben, you were thinking of other than
No, that's good. Thank you.
yeah? Yeah, so that's just a really useful thing you can do in Jupyter Notebooks where and that will let everyone see how exactly your plots were generated, and they can reproduce that exact plot on their own computer just to see that, your stuff is reproducible. So we have a couple more Q&A questions. How do we decide what genome assembly we should use? For most species, by default, I would go to NCBI and pick the reference genome from there. If you have your own genotype, but you have a genome, you might want to use that. this can get tricky for, I think, a couple of species like maize, where there's a couple different versions would have different pros and minuses. I know a lot of people for maize are still on V3 instead of V4 or V5, which is what we're up to now. So that can be quite tricky. It really just depends. We're gonna wheat, yeah, wheat's that's a tough one. Um, I mean, it's gonna, yeah. I mean, there's no real good answer like for knowing exactly which one to pick. But uh, if you're having issues with alignment, sometimes the NCBI ones can be weird, like have issues, and it might be easier to get it from the species um, specific site. So like maize GTB or tear or the rice database, those can sometimes be like maybe a bit easier to use if you're having issues with like the download or the setup as well. How do you recommend choosing clustering resolution in practice? That's a great question. There's a couple of manuscripts on methods to try and tell if your clusters have heterogeneity that should be further subclustered. There's not really a good answer. This is why I've really tried to emphasize that you need to be involved in the data and like really understand what's going on in the data, because there clustering resolution applies to the whole data set. So there are functions in ScanPy and Seurat for subclustering a specific cell type. So if you have one cluster that you think needs to be subclustered, you can subcluster only that one. But yeah, for that you need to really just look at your data, because there's no good way to pick a resolution a priori without having played with the data. How do you approach learning a new technique? I think really the way to go is to just actively trying to do something with the data is the best way to learn for this kind of data analysis or programming. Because really just actually handling the data and manipulating it and trying to find things is just the best way to gain experience. When I started my PhD, I had no dry lab experience. I'd done purely wet lab work. I ended up doing a fully dry lab PhD, just analyzing data and trying to gain just through hands-on dirty experience. Yeah, so I would definitely recommend that more than just reading tutorials forever, because at the end of the day, manipulating the data and manipulating is the wrong word. Handling the data and trying to make inferences and making mistakes and errors is just the best way to learn. Yeah. Yes, it's possible to download directly to a Linux environment if it's too slow on your laptop. If you open at home, you'd use something like the curl or wget command to do that kind of a download directly in the Linux environment without having to do like download and then SCP it onto your data. So you'd on a website, you'd right click and you pick copy link address, and you do like curl and then paste that link address to download directly onto a Linux environment, typically. There's also, if you're getting that from SRA, there's SRA tools, which is a array to download directly from SRA. That's quite quick to download it directly to the Linux environment. Your opinion on how many biological replicates one should generate for a single cell analysis? Great question. Uh, I would say personally 3 batches is how many you should generate. I think there's probably some people who have an opinion more like 6, but I think 3 is really the minimum for batches that you'd like to do. I mean, this also depends on if you're trying to do a pilot experiment versus an analysis. It really just does depend, on what you're trying to do or what you're trying to show. But having 3 batches is helpful. Okay, I guess if we don't have any more questions, I think it might be time to wrap it up. One more question before we wrap up. It's a question on integration, which is always a very tricky topic. Question is, when working with samples that correspond to developmental stages and contain different sets of types, should all the samples be integrated, or is it better to integrate the replicates for 2 conditions separately? If your replicates are different developmental stages, I would personally integrate the developmental stages separately instead of integrating everything. But it's a very good question because the answer is it's quite tricky. I think typically you should try and integrate minimally as possible, well, to make your data like comparable, because you can lose a lot of real biological variation in integration. But you do need to integrate often to be able to make any comparison. So yeah, I would try and do the minimum integration possible. So I'd start with just integrating at the development stage level and then integrate further if necessary. Yeah. Ben also mentioned rendering as a PDF. That's a great point just in general. Uh, um, uh, these notebooks can be output both as scripts, so you can just click a button to output it as a script, which will a Python script, which is quite useful, or you can render the whole thing as a PDF, so you can save it as a document that you can share with people. That's it's not interactive, so it will show what do you want to do. How much should be the cutoff for read aligned, if any? So that's referring to like cutoff alignment. I mean, it just depends on how much on your specific sequencer workflow. So this is going to just really depend on your workflow. We're going to single nuclei RNA from data from different conditions. So I have multiple biological replicates per condition. How do I integrate these samples into ScanPy? I would I would integrate biological replicates per condition first, just across biological replicates, and see if the data looks reasonably integrated without any further integration. And if not, I would then try integrating all of the data sets at once across replicates to see if that is better. So I would start with integration of so ScanPy has a bunch of integration methods built in. I like Harmony quite a bit. It's not super aggressive, but and for biological, it's quite good. I find if you have like different species to integrate, then you might need something more that's a bit more advanced, like SCVI tools. But yeah. Um, Great. Is there anyone else on the panel want to mention or include about Jupyter notebooks or reproducibility or anything?
Just one thing that came to mind for me when the question about reference genomes was being discussed. It's often the case that if you get a reference genome, it won't include organelles. So if you do go to NCBI, they will provide kind of 2 versions, one of which is called the RefSeq and one of which is called the GenBank. And usually the only difference between those two is that NCBI has added organelles to the RefSeq version so that it's complete. And those might come from like a different genotype, the same species, but it's good to include the organelles so you can have the QC.
Great point. Yeah. Okay, great. Well, thank you guys for all your great questions. It's really all good stuff to think about, especially integration. I've done a lot of my PhD was focused on it, so there's a lot of open questions in the field, but yeah. Um, great. I'll stop sharing and
Thank you so much. I do just want to add a couple of points real quick, that we will have all of the recordings from the 3 days posted onto the PCA YouTube channel. And we will send out an email that includes the links to those YouTube videos, as well as the certificates that say that you have completed this workshop. And we will also include an exit survey, just kind of asking for your opinion on how this went and how we can help you with other workshops in the future. And that will all be coming within the next week or so once we can get those videos uploaded. The last point that I do want to make is that I will also include the link to the Padlet, which has all of the questions that we have asked, including some of the written responses. And I will also include, if any more questions come in now, I will include those over there. And then speakers, if you want to go ahead and go over and check out the Padlet and answer any unanswered questions or add to any of the answers that are already there, that would be really great, but not a requirement. But yeah, thank you everybody so
yeah.
much.
Yeah, I'll answer really quick some of these methods. For pseudobulking metacells, there's literally metacells, which is great, but pseudobulking is very simple. And you can do that like pretty easy. Like the code I used is like a very basic pseudobulking. It's also pseudobulking by clustering at a really high resolution. So you run the Leiden algorithm at a really high resolution to get about 10 cells, 10 to 15 cells per cluster, and then just merge every cluster. That's something that approach I've used that's quite very easy to do, very quick, and very effective. And then what criteria do you use to determine whether pseudotime reflects true developmental progression versus an artifact of dimensionality reduction? That's a great question. If you have an answer, I'd recommend publishing it. Uh, seriously, I mean, you just need to use whatever true developmental, uh, uh, like true developmental timeframes you have available to try to get an idea of it. But I personally am not a huge believer in pseudotime for plant data. I've tried to get it to work for literal years on some data sets with stage data, and we never really got it working. This is, I think, related to plants' lack of terminal differentiation. But in plants, I really do not think we have good pseudotime methods that work well.
So I just wanted to say just a couple of things. I wanted to thank all of the speakers and panelists that have participated and really, spent a lot of time developing this workshop. I think it's gone really well. I'd also really like to thank everyone who's attended. And I'd especially like to thank Joanna for organizing this whole thing and setting up all of the logistics and infrastructure. I think she's done a great job and really kudos to her. And I think with that, we hope this was a helpful seminar series. And if you have any questions or want to reach out, I think you guys know where to find us. So thanks.
Great. Thanks, everyone.